Hello, my name is Eric and I'm here to present this study that's part of my PhD, and it was named Variation Partitioning of Local and Geographic Factors in Structuring Bacterioplankton Metacommunities using High Throughput Sequencing. The biodiversity was one of the most studied topics of ecology in the last decades. Distinct organisms in different natural contexts were already studied, and thanks to this, we know now that the degree of species dissimilarity between two sites is mainly dependent on the balance between the site's isolation, environmental heterogeneity, and the dispersal capacity intrinsic to each organism. Distinct statistical methods were already created to reach the biodiversity. Those methods can be grouped in two main types by the form in which the data is considered. Some need species identity data, which is the information about the presence or absence of each species in each site, and is especially useful to analyze biodiversity changes and decision-making for species conservation. Others need the abundance, which is important information to observe ecological aspects. Another statistical method that was being extensively used is the variation partitioning. This method is especially useful as a complement to the biodiversity because it permits to measure and rank the local and regional characteristics that are shaping the observed dissimilarity. Bacteria are organisms which present great dispersal capacities and remarkable diversification and adaptability. By these characteristics, it's not surprising that they present a huge biodiversity, and distinct studies have been concluded that this biodiversity is mainly shaped by the environment. But the advance of culture-independent methods showed that the microbial biodiversity was much higher than it was supposed to be. Also, few studies applied a more complete approach to observe all the geographic aspects that can impact the bacterial biodiversity in natural systems. In this sense, our main objective in this study was to investigate the impact of spatial and environmental characteristics influencing the bacterial dissimilarity between shallow lakes scattered over a great landscape. We also took the opportunity to analyze the impact of different types of biodiversity measurements. So, we hypothesized that first, the observed biodiversity should be huge and reflect great influence of space turnover on this metacommunity, caused by environmental heterogeneity and different degrees of isolation by distance. Second, the dissimilarity between sites should be mainly impacted by the environment, followed by the spatially structured environmental factors, and finally by the geography. And third, these two predictions should be more easily observed when the abundance are considered. To this study, we selected 60 samples in a sampling effort that covered an area of nearly 250,000 square kilometers and 4 distinct geographic regions. In each shallow lake, environmental and biological samples were recovered. The bacterial DNA was sequenced and the taxonomy was obtained as amplicon sequence variants, or ASVs. The ASVs data were rarified and transformed into dissimilarity matrices. For this, in our analysis, when it was necessary, we applied the Jacquard approach to the species identity matrices and breakers to the abundance ones. Then, the dissimilarity matrices were used to conduct the beta diversity partition and the half-creek index. After, we prepared the environmental and geographic data to variation partitioning. So, the environmental data was standardized, and after that, we conducted a redundancy analysis, followed by an ordination test to select the factors that were in fact linked to the biological data. And after that, a collinearity test was applied to see if any part of these selected variables are collinearly impacting the data. Finally, all the variables that passed these checking points were selected from the standardized file to compose the environment fraction. For the geography, we applied a distinct engine vector approach to reach all the spatial characteristics that can be impacting this metacommunity. First, we used the DBMan approach. In this approach, that is generally used to infer the impact of geography on bacteria, all sites are considered as equally connected in the landscape. Second, we used a man approach combined with weight information. In this case, we considered that the increasing distance should difficult the connection between sites. So, the distant sites have a weaker connection than nearby ones. And finally, we used the man approach, but here we considered that all sites from the same geographic region are closely related, while the sites from distinct regions are weaker connected, even when close to each other. After that, we conducted the same steps from the environment fraction to obtain the geographic fraction. And finally, we applied the variation partition to see how much these fractions are connected to dissimilarity matrices. The beta diversity partition showed a great dissimilarity between the sites for both species identity and abundance data. It was expected and had been extensively shown in the literature. It's interesting to point that the species identity, that is the primarily used approach, show an increased importance of nestedness when compared to its equivalent for abundance data. The half-creek results showed that the species identity approach results in a major part of pairs of sites more similar than expected by chance, while the abundance showed that the major part of sites present no significant difference or a greater dissimilarity than expected by chance. These results evidenced one time more that the abundance is a quite important variable when analyzing bacteria, and the greater part of dissimilarities, and consequently the process where these organisms are important, are impacted more by difference in local abundance than the absence of one or a group of key organisms. Finally, there is the variation partitioning. Our data showed a great fraction of no explained variation. This pattern may be explained by the stronger isolation of our samples that led to an increased importance of local variability and a decreased regional synchrony. In this analysis, the abundance alone had reduced the unexplained fraction by about 10%, which indicates that this unexplained fraction can be also caused by one important and not measured information, maybe an environmental factor. Another possibility is that some variable temporarily structured is impacting the metacommunity, but it needs to be better explored in the future. In general, geography is the best predictor of variance for our data. We hypothesized that it can be explained by the interaction of two variables. First, our dataset is composed by sites with higher isolation, and it's natural to expect that the geographic factors will have increased importance for the dissimilarity. Second, we used two approaches further the commonly adopted to infer the geographic importance. So, we may have reached other variables not previously observed, but that in fact can affect the bacterial dissimilarity. As a complement, we partitioned the pH to all other environmental variables to see that this factor alone is responsible by 1/3 of variation explained by the environment. This was not surprising because the importance of this variable to bacteria was shown before in other works. Finally, the partition of geographic fraction showed that the region is the best predictor of dissimilarity, while the space, the approach most commonly used, is the worst. These results reinforce the idea that the methodological refinement from this study was important to reach these results. So, to conclude, in this study, we contributed to reinforce the general view that, despite the importance of species identity datasets to infer biodiversity and contribute to decision making, for bacteria, the abundance is an important variable, and it can lead the observer to quite different conclusions about the bacterial biodiversity in a region of interest. Also, the geography can be a great predictor for bacterial dissimilarity, spatial in cases of higher degrees of isolation. And it's recommended to adopt a more complete approach aiming to grant that the geographical aspects are being considered. So, it's all I want to tell about this study. I hope you enjoy it. Thanks for watching.